After studying this module, you shall be able to classify the different types of nitrogenases, know the bacterial species containing those enzymes, identify the difference and similarity between the nitrogenases, learn how molybdenum nitrogenases is better than others, know the limiting stoichiometries of molybdenum, vanadium and iron-only nitrogenases. Introduction. 4 types of nitrogenase enzymes have been characterized so far, out of which 3 of them are homologous enzymes, are genetically non-identical but have similar metal cofactor and protein subunit composition. These are the molybdenum nitrogenase, vanadium nitrogenase and iron-only nitrogenase. The molybdenum nitrogenase is the most commonly distributed nitrogenase in nature containing the iron-molybdenum cofactor. It is also considered to be the most efficient enzyme for nitrogen fixation. The paradigm that biological nitrogen fixation is dependent on molybdenum was established more than 60 years ago, which prevented the investigation of other clusters related to this process. The evidence for the existence of non-molybdenum nitrogenases were therefore often disregarded. But soon the situation changed when 2 molybdenum independent nitrogenase systems were discovered and isolated in Azobacter vanillii. One is a vanadium dependent nitrogenase that is formed only under molybdenum deficient conditions, and the other is iron only nitrogenase that is formed under extremely low concentrations of both molybdenum and vanadium. An exciting new terrain of research on nitrogenase started with these alternative nitrogenases. Till date, the V nitrogenases from A. vanillii and A. croceucum and iron-only nitrogenases from A. vanillii, Rhodobacter capsulatus and Rhodospirillum ruberum have been isolated and biochemically characterized. These alternative nitrogenases may perhaps have technological applications owing to their high rates of hydrogen formation by proton reduction. Although most diisotopes have only the molybdenum nitrogenase, but some of them also synthesize alternative vanadium nitrogenase and or iron-only nitrogenase enzymes containing the iron-molybdenum cofactor like iron-vanadium cofactor or iron-iron cofactor metalloclusters at their active sites respectively. Interestingly, there are no reports of diisotopes lacking in molybdenum nitrogenase and carrying exclusively an alternative nitrogenase. Regulation of nitrogenase expression in bacteria carrying alternative nitrogenases is dependent on the availability of molybdenum, vanadium or iron in the medium. The molybdenum containing, vanadium containing and iron only containing nitrogenases are not equally distributed in nature. Some organisms like Azobacter vanillii possess all 3 types of nitrogenases, while other organisms like Rhodobacter capsulatus and Rhodospirillum ruberum have the molybdenum containing and iron only nitrogenases. And some organisms like Enebena verbilis carry both molybdenum and vanadium containing nitrogenases. Besides this, Klebsiella pneumoniae possess only the molybdenum containing nitrogenases. Additionally, a 4th type of nitrogenase has been recently discovered which is not phylogenetically related to the above ones. This nitrogenase is found only in Streptomyces and carries a molybdenum molybdopetrin cytosine dinucleotide cofactor as an active site and exhibits entirely different protein composition. Different types of nitrogenases enzyme. Molybdenum nitrogenase. The molybdenum nitrogenase is a complex enzyme containing an active site where a molybdenum iron sulfur metallocluster comprising an inorganic Fe6S9 core coordinates a central light atom X, which can be carbon, nitrogen or oxygen, and is capped with external iron and molybdenum atoms. The molybdenum atom is coordinated by a molecule of R homocitrate through its C2 carboxyl and hydroxyl groups. This has been represented in figure. The role of the molybdenum subsite in iron molybdenum cofactor is unclear. The usual molybdenum containing nitrogenases that found in all diisotopes consist of 2 independently purifiable protein components, molybdenum iron protein and iron protein. The iron protein is a homodimer containing an Fe4S4 cluster is responsible for electron transfer to the iron molybdenum protein in an ATP dependent process, whereas molybdenum iron protein exhibit an alpha 2 beta 2 tetrameric structure and contain two pairs of metal centers, the P cluster and molybdenum iron cofactor centers. The detailed structures of the A-vanillin and Clostridium pastorinium molybdenum iron proteins have been revealed by X-ray crystal structure analysis. The P cluster seemingly transfer electrons to the molybdenum iron cofactors. The molybdenum iron cofactors are certainly the site of dinitrogen binding and reduction. Also, the recent crystal structure of the iron protein and the molybdenum iron protein complex supports the above described electron transfer scheme. From the crystallographic analysis, it was found that iron molybdenum cofactor is located in the alpha subunit and is covalently attached to the protein by a cysteine and a histidine residue. Besides, several hydrogen bonds exist between the proteins and various sulfur ligands of the cofactor. Crystallography and EXAFS have identified two characteristic iron-iron distances. One is relatively short, 2.6 Armstrong, which is assigned to the iron-iron nearest neighbors, and the other one is long, 3.7 Armstrong, assigned to the vectors across the prism. The P cluster is found in between the alpha and the beta subunits and is linked by 3 cysteine residues to each subunit. The structure and mechanism of the molybdenum nitrogenase has been discussed thoroughly in the previous modules. The structure and mechanism of molybdenum nitrogenase enzyme. V-nitrogenase. After 50 years of the role proposed by researchers for vanadium in biological fixation, it was demonstrated that in addition to their well-characterized molybdenum nitrogenases, there is genetically distinct nitrogenase system in both Azobacter vanillini and Azobacter crocugum, where the conventional molybdoprotein is replaced by vanadoprotein. Both these nitrogenases system require similar conditions for their activity, such as MgATP, a low potential reductant, and the absence of oxygen. Vanadium nitrogenase is an alternative key enzyme found in nitrogen fixing bacteria and is employed for nitrogen fixation. When molybdenum is unavailable, that is, the genes encoding the vanadium nitrogenase are expressed only under molybdenum deficient conditions. Since most of the functions of vanadium nitrogenase match with those of the more common molybdenum nitrogenases, they therefore serve as an alternative pathway for nitrogen fixation under molybdenum deficient conditions. It is therefore considered to be an important component of the nitrogen cycle that converts nitrogen gas to ammonia, thereby making otherwise inaccessible nitrogen available to plants. V-nitrogenases are found in species of bacterial genus Azobacter as well as in Rhodosudomonas palustris and Anemona verbalis. V-nitrogenase of A. crocugum is composed of a tetrameric vanadium iron protein with a molecular weight of 210,000 with an alpha 2 beta 2 structure containing 2 vanadium atoms, 23 iron atoms and 20 acid labile sulfide atoms per tetramer, and a dimeric iron protein with a molecular weight of 64,000 with a gamma 2 structure containing 4 iron atoms and 4 acid labile sulfide atoms per dimer. Vanadium K-edge X-ray absorption spectroscopy illustrate that vanadium in the vanadium iron protein is very similar in molybdenum iron protein, having sulfur, iron and possibly oxygen as nearest neighbors. Although vanadium nitrogenase has an alpha 2, beta 2, gamma 2 subunit structure, while molybdenum nitrogenase has an alpha 2, beta 2 structure, a vanadium and iron containing cofactor can be extracted from the vanadium iron protein and can reduce carbon monoxide to ethylene, ethane and propane, unlike molybdenum nitrogenase. But both these enzymes can reduce protons to hydrogen gas and acetylene to ethylene. The products of C2H2 reduction by the hybrid protein are a characteristic of the vanadium iron protein and provide evidence that iron vanadium cofactor forms part of the active site of vanadium nitrogenase. Like molybdenum nitrogenase, H2 functions as a competitive inhibitor And carbon monoxide functions as a non-competitive inhibitor for nitrogen fixation. Although the structural genes encoding vanadium nitrogenase show only about 15% conservation with molybdenum nitrogenases, the two nitrogenases share the same types of redox centers. V nitrogenase is less effective at fixing nitrogen than molybdenum nitrogenases under room temperature because it converts more H plus to H2 as a side reaction. However, at lower temperatures, vanadium nitrogenases have been found to be more active than the molybdenum type, and at temperature as low as 5 degrees centigrade, its nitrogen fixing activity are 10 times higher than that of molybdenum nitrogenase. Like molybdenum nitrogenase, vanadium nitrogenase can be easily oxidized and is thus active only under anaerobic conditions. Various bacteria employ complex protection mechanisms to avoid oxygen. The overall stoichiometry of nitrogen fixation catalyzed by vanadium nitrogenase can be summarized as follows. N2 plus 12 electron plus 14 protons plus 24 mg ATP gives rise to 2 ammonium ions, 3 H2, 24 mg ATP and 24 HPO4 2 negative. Research on vanadium nitrogenase demonstrated that it possesses the ability to reduce carbon monoxide into trace amounts of ethane, propane and ethylene in the absence of nitrogen via dithionate and ATP hydrolysis. The process of formation of these hydrocarbons is conducted through proton and electron transfer, where small carbon chains are formed and might allow the production of hydrocarbon fuel from carbon monoxide at an industrial level. While the majority of X-ray spectroscopic studies have been performed on molybdenum dependent nitrogenases, there has also been recent progress on studies of vanadium nitrogenases by the collaborative groups. The differences between molybdenum and vanadium dependent nitrogenases are of particular interest since the latter has proved to catalyze carbon-carbon bond formation effectively. This difference in reactivity could be probably due to the differences in the electronic structures. The earliest XAS studies on vanadium nitrogenase from Azobacter procucum were carried out by Erber et al. In 1987, and it was concluded that vanadium site had a distorted octahedral environment with an oxidation state somewhere between 2 and 4. Later in 1988, George and co-workers found an elongation of metal iron distances in vanadium nitrogenases relative to molybdenum nitrogenases, which they indicated could relate to differences in observed reactivity. Thus, the differences in the electronic and geometric structures of iron molybdenum cofactor and iron vanadium cofactor have opened up research opportunities for further investigation. Iron nitrogenase. The existence of an iron-only nitrogenase system was found only when the ambient molybdenum concentration is extremely low, as demonstrated in R. capsulatus. Recently, a rapid and gentle isolation procedure yielding highly active protein components, iron-iron protein and iron protein, has been established along with the extensive investigation on the biochemical and EPR spectroscopic properties of this enzyme. The structural information on the metal clusters of iron-only nitrogenases by spectroscopic methods is still lacking. Nevertheless, there are genetic and biochemical indications that the iron-iron cofactor has very similar structure as that of molybdenum iron cofactor and iron vanadium cofactor, the structure of iron-only nitrogenase. Results obtained from the spectroscopic, biochemical and genetic investigations of nitrogenase system revealed that iron molybdenum cofactor and heterometal free cofactor of iron nitrogenase are structurally homologous. The only fundamental structural difference between the two cofactors is an additional iron atom replacing the molybdenum atom in the iron nitrogenase cofactor. The findings substantiate that designation of cofactor of the iron-only nitrogenase in R. capsulatus must be iron-iron cofactor due to its structural homology with the iron molybdenum and iron vanadium cofactors. Binding and reduction of nitrogen can also be catalyzed based solely on iron by iron-only nitrogenases, which is so far largest member of the family of naturally occurring iron sulfur clusters. The fourth type of nitrogenase. The fourth type of nitrogenase is a molybdenum nitrogenase, phylogenetically that is unrelated to the other three classes of nitrogenase enzyme and has only been found in Bacterium streptomyces, and this molybdenum nitrogenase exhibit completely different biochemical features consisting of different protein composition, insensitivity towards oxygen, low requirement of MgATP and molybdenum cofactor type of cofactor at the active site. How molybdenum nitrogenase is better than the rest? Among all the three nitrogenases, molybdenum nitrogenase is the most efficient in terms of nitrogen fixation owing to less waste of energy. Limiting stoichiometries are as follows. Initially, molybdenum was considered as the most likely site for substrate binding and catalysis, since molybdenum nitrogenase is significantly more active in nitrogen reduction than its vanadium or iron analogues. Moreover, molybdenum compounds can readily coordinate nitrogen and its reduced forms. However, recent combined spectroscopic and substrate inhibitor binding studies strongly indicate that the iron sulfur X core is the initial substrate binding site, whereas current mechanistic models do not include a direct function for the molybdenum subsite. Summary. Nitrogenase is a complex metalloenzyme that catalyzes a key step in the global nitrogen cycle, the reduction of inert dinitrogen to bioavailable ammonia. Three homologous nitrogenases have been identified which resemble one another in primary sequence and cluster type: molybdenum, vanadium and iron only nitrogenases. All of them are two component system comprising of iron protein and molybdenum iron, vanadium iron and iron iron protein. All these are distributed in group of symbiotic and asymbiotic bacteria. There is a fourth type of nitrogenase known as molybdenum nitrogenase, phylogenetically which is unrelated to the other three classes of nitrogenase enzyme found in Bacterium streptomyces thermoautotrophicus. This molybdenum nitrogenase exhibits completely different biochemical features consisting of different protein composition, insensitivity towards oxygen, low requirement of MgATP and a molybdenum cofactor type of cofactor at the active site. Of all, molybdenum nitrogenase is the most investigated enzyme and most efficient in terms of nitrogen fixing owing to less waste of energy.